All right, so you guys made it. This is the last experiment, the last lab, experiment 19, the aspirin synthesis and analysis. Remember, this is a full lab write-up, just like the last two you guys have done. All right, so I'm going to go over some quick background information on aspirin. You guys can use this information and put as much as you can into the intro section for your guys' full lab write-up. All right. So, throughout history, many compounds obtained from nature have been used to cure illnesses or to produce an effect in humans. These natural products have been obtained from plants, minerals, and animals. In addition, various transformations of these and other compounds have led to even more medically useful compounds. That leads us to acetylsalicylic acid, also known as aspirin, which is a synthetic organic derived from salicylic acid. Salicylic acid is a natural product found in the bark of the willow tree and was used by ancient Greeks and Native Americans, among others, to counter fever and pain. However, salicylic acid, the natural product, is super bitter and irritates the stomach. Thus, acetylsalicylic acid is what we use. All right, so analgesics are compounds that are used to reduce fever, and antipyretics are compounds used, analgesics are compounds used to reduce pain. Antipyretics are compounds used to reduce fever. And then a popular drug that does both is aspirin. So aspirin particularly blocks an enzyme called cyclooxygenase, here shown as COX-1 and COX-2, okay? That's the enzyme that it blocks. The acetyl group, right here highlighted in blue, on the aspirin gets hydrolyzed, and then the acetyl group gets bonded as an ester to the alcohol group of the serine residue on these enzymes. Okay. Let's talk about prostaglandins. So prostaglandins are unsaturated carboxylic acids consisting of a 20 carbon skeleton. They also contain a 5-member ring and are based upon the fatty acid arachidonic acid, which is just seen here. One of the main functions of prostaglandins include activation of inflammatory responses. So they basically produce pain as well as fever. So when a tissue gets damaged, white blood cells flood to the damaged site, try to minimize tissue destruction, and prostaglandins are produced in that site as a result. So we know that prostaglandins induce inflammation, pain, and fever. But as was said earlier, aspirin blocks the enzyme cyclooxygenase COX-1 and COX-2, which these enzymes are involved with the ring closure and addition of oxygen to arachidonic acid, converting the arachidonic acid to prostaglandins. So what aspirin essentially does is inhibits the enzyme COX-1 and 2 that converts the fatty acid arachidonic acid in your body into prostaglandins, reducing inflammation. So by inhibiting or blocking this enzyme, the synthesis of prostaglandins are blocked, which in turn relieves some of the effects of pain and fever. All right, that's basically just a little bit of background on aspirin. You guys can use it after your intro. Now let's get into what I want to see in your guys' full lab write-up. So remember that this is your last lab and it's full lab write-up. But before you guys even get started on the lab, you guys can and should have the title for this lab already written up, along with the source, which is just citing the lab manual, as well as the purpose, which is to synthesize and determine the purity of aspirin. And then regarding your introduction, I want you guys to make sure to include the chemical name of aspirin. Include that aspirin is composed of an ester and an organic acid. Include its function. This is all going into the intro. Include the difference between salicylate ion and salicylic acid. Also include what type of acid the salicylic acid is. Is it a strong versus a weak acid? And then I want you to include how to determine the purity of aspirin, and don't forget to include the chemical equations that you guys are going to see in your lab manual into the intro. And then here are some of the chemical equations that I want to see in your guys' intro. And then here is basically the answer. Aspirin is a weak monoprotic acid. I'm sure that's in your guys' lab manual. All right, so moving on to the procedure and data section. So for part A, there's 3 parts of this lab: part A, B, and C. Part A is all about the synthesis of the aspirin. Part B is basically checking the melting point of the aspirin. And then part C is the titrations. So for part A of the procedure section of this lab write-up, you guys should write in your procedure column that you need X amount of grams of salicylic acid, 4 to 5 mils of acetic anhydride, 5 drops of concentrated sulfuric acid, which you'll see in the video combined and heated for like 5-10 minutes. And then you'll also see in the video that the heat source is removed, 10 mils of DI ice water is added to decompose excess acetic anhydride. Then you're going to see the solution is going to be chilled in an ice bath until crystals stop forming. So that's all going on the left side, which is the procedure side of the column. For the observations column, which is the right side, make sure to include the molarities and appearances of the reagents that were used. So like for sulfuric acid, you're going to put it is a blank colored solution, and we use a blank molar concentration, etc., etc. For like salicylic acid, you're going to say they are blank colored crystals. Include also what is dissolving, as well as the solutions, like if there's any precipitate in the solution, stuff like that. All right, so continuing with part A, in the video, you're going to see a vacuum filtration setup. What you'll see is 10 mils of ice water added to the flask. Then the crystals will be transferred onto the filter and eventually dried. Then the crystals will be washed with 10 mils of ice water, and then they will be transferred to a beaker where ethanol will be added to the beaker, which contains the crystals. Then the mixture will be warmed up, cooled, and vacuum filtered once again. So for your observations, note what ethanol does to the crystals, note any cloudiness in the solutions, and note when the crystals reappear. Okay. All right, in regards to your data section, make sure that in your lab write-up for your data section, include the table containing the mass of the weight paper, the molar concentration of sodium hydroxide solution, the burette readings, including the initial and final readings, the moles of acetylsalicylic acid used, etc., etc. You're going to see a table like that in your lab manual for trials 1, 2, and 3. I want you guys to include that and fill it out in your data section. And then make sure to include a separate calculation section that shows the calculations for everything in the table for one of the trials, not all the trials, just one. And then the calculation section, once again, should be separate from your data section. All right, so moving from part A to part B. Part B, the melting point, we're going to check the melting point, and what you're going to see is a capillary tube, something like this, filled with some of the aspirin crystals, right? And then this capillary tube is going to be placed into a melt temp that looks something like this, where it will be heated, and then the crystals will eventually melt. So you're going to see a total of 3 trials done for this melt temp of the aspirin crystals. And then make sure to note in your observation section the appearance of aspirin when it starts to melt, when it is almost fully melted, and then also when it is completely melted. And then remember that the melting range is defined as the range of temperature from the point at which the crystals first begin to liquefy, all the way to the point at which the entire sample is completely liquid, okay? So you're gonna get a range of temperatures for the melt temps. All right, moving on to part C, you'll have to calculate the mass of aspirin needed for 20 mils of 0.1 molar sodium hydroxide to reach its stoichiometric point, right? You guys just basically did that. Then you'll see in the video that the mass of aspirin is measured out and placed in a flask. I think the masses of the aspirin is actually going to be emailed to you guys via the head TA, Peter Weiner. So you guys should have an email already with the randomly generated data that you guys are going to need. But anyways, so part C, you're going to see 10 mils of 95% ethanol, 50 mils of DI water, and 2 drops of indicator added into the flask that contains the aspirin crystals for part C. So yeah, the indicator, the phenolphthalein indicator is in there, and then now you should prep, now you're going to see in the video that your burette is going to be prepped with the base, the sodium hydroxide, the 0.1 molar sodium hydroxide. So I want you guys to note the volume of the initial reading of the burette, right? I'm watching this video, just note the initial reading. And then in the video, then you'll see the titration start to begin. And then as you're watching it, note the volume of the burette after the solution remains like light pink, after it's reached its endpoint. So in the video, the titration will run past the endpoint. So it will be over titrated. So it's up to you guys to choose the correct endpoint and obtain the final burette reading at that endpoint that you choose is the correct one. And then the rest of the data needed to finish this part of the lab is individually generated and sent to you guys via email. All right, now we're at your results, and now we're going to talk about your results and errors. So this is going to get kind of wordy, so you guys should pay attention, but we're going to talk about the percent yield. So remember that the percent yield is just the actual yield over theoretical yield times 100%. So that's pretty simple. And then so in terms of theoretical yield, you should have your theoretical yield from the pre-lab assignment or pre-lab quiz that we just took. But in case you didn't get it, what you want to do is you want to take the grams of salicylic acid multiplied by the mass, by the molar mass to get moles, right? Then you multiply that, sorry, one second. Then you multiply that by the ratio of the moles of acetylsalicylic acid over the moles of salicylic acid, which is a 1-to-1 in terms of stoichiometric ratio. That leaves us here. And then you multiply the moles of acetylsalicylic acid by the molar mass of acetylsalic acid to get the grams of acetylsalic acid, which is going to be your theoretical yield, okay? All right, so let's talk about percent purity. To get the percent purity, you should know how many grams of acetylsalic acid reacts with 20 mils of 0.1 molar sodium hydroxide from the pre-lab quiz or the pre-lab assignment. But in case you didn't get that, what you gotta do is you gotta take the volume of sodium hydroxide times the molarity of sodium hydroxide to get the moles of sodium hydroxide, right? And you take the, and since the moles of sodium hydroxide to the moles of acid is 1-to-1, you basically have your moles of acid. Then you times that by the molar mass of acetylsalic acid to get your grams of acid in the sample. And then you take the grams of acid divided by the theoretical yield times 100% to get the purity. Let's see, the average melting temp you get by taking your 3 melt temps, add them together, divide by 3. Pretty simple. Okay, let's continue. So your conclusion section, what you guys want to include is your percent yield, your average melting point, the average percent purity of aspirin, and that's pretty much it. All right, let's talk about some closing thoughts. So today, like I said, is the last Zoom class meeting. In terms of grading, what I have to grade for you guys is Dry Lab 3, Experiment 35, and Experiment 36. So those labs should be graded before Sunday of this week. And then once you guys turn in your full lab write-up for Experiment 19, I'll grade that on Monday, and then grading should be done. Let's see. And don't forget about your lab simulation. It's called Organic Chemistry Introduction. You also have that due as well. All right, that's basically it. If you guys have any questions, I'll stick around. If not, you guys take care.